All right. Hello, everybody. My name is Meredith Wardell. I am one of the faculty here at the paramedic program. I'm also the clinical coordinator for our clinical internship, and I'm also the paramedic prep coordinator. I wear a lot of hats around here. But today we are going to be talking about the cell structure and cell function, and an overview of the smallest functioning unit of life, which is a cell. Starting off with cytology, it's the study of cells. Cells are the smallest units of life to be considered alive, and they adapt to different functions and environments. Cells come in all shapes and sizes. Their structure often matches their function. So, nerve cells are long and thin so they can transmit signals over long distances, and muscle cells are shaped for muscle contraction. Even red blood cells, they have like a donut-like shape to them to help carry oxygen efficiently. When similar cells work together, they grow close and they form tissues. Think of it like a team. Each player represents a cell, and each cell does a specific job, and together they make a team. They make something bigger. All living things are made up of cells. Every function of life begins at the cellular level. Every complaint, injury, illness, infection that we see as healthcare providers can all be stripped down to its most basic layer, and that's a cellular issue. Cells can grow, reproduce, they respond to their environment, and they use energy. And if something can do all of that, we consider it to be a living organism, even if it's just one microscopic cell. So not all cells are the same. They all share certain characteristics, but they have key differences in multiple different things, including whether or not they have a nucleus. Eukaryotic cells contain a nucleus, and as you can see in this image, genetic material is enclosed within that nucleus, and then prokaryotic cells do not have a nucleus, and their genetic material is not enclosed into one specific location, but instead it freely resides within the cell. Instead of a nucleus, there is a nucleoid, which is a name derived from nucleus-like, because it serves a similar function to a nucleus, but does not have the same structural complexity that a nucleus has. So a single cell, the zygote, develops into a complex multicellular human being. We were all at one point a zygote. It is the very first cell of a new life. It forms when a sperm cell fertilizes an egg. It contains all the genetic information needed to create a human being, half from the mother and half from the father. Going from zygote to embryo, the zygote divides through a process called mitosis. It becomes like a ball of cells, like you see here. And it eventually forms a structure called a blastocyst, which implants into the wall of the uterus and starts developing an organism called an embryo. The cells begin to specialize or differentiate to perform different functions. Even though they all started out the same, the cells start to figure out what their job is, what their role is in this process. By about 8 weeks, that embryo becomes a fetus. And this stage is marked by the development of organs and body systems within the fetus. However, cell division continues. The cells are now forming, except the cells are now forming specific tissues and organs. And then around 9 months of development, the fetus is fully formed and ready for birth. But what's so incredible is that the fetus just started out as a single cell, a single zygote. We all were one once. But I think this is a great way to show how cells work together to form something so much bigger. We have over 30 trillion cells in our body. Let's just pause and take that in for a second. 30 trillion cells, which is a huge number. I can't even imagine what that number would look like. But to help you picture it, If you lined up 30 trillion cells end to end, it would stretch over 744,000 miles, which is enough to wrap around the Earth 30 times. And all of that fits into our body. That's the level of microscopic complexity that we're dealing with. It's so small that we have 30 trillion in our body. And the cells don't just sit there. They're constantly being replaced. Skin cells live for a few weeks. Red blood cells live for about 120 days. Some cells, like nerve cells, can last a lifetime. But overall, for the most part, our bodies are repairing, growing, and responding to changes by making new cells. All right, so let's walk through the life cycle of a cell. This is the process that it goes through to grow, divide, and specialize. This cycle is essential not only for growth and repair in multicellular organisms like us, but also for reproduction in single-celled organisms. So the first phase is interphase. Most of a cell's life is spent in interphase. This is where it's growing, it's performing its normal functions, and it's preparing for division. And in this stage, most importantly, DNA is replicated. The cell makes an exact copy of DNA before it gets ready to divide. In the next stage, which is called cell division or mitosis, is the process of dividing the nucleus. So during mitosis, the duplicated chromosomes are carefully separated into two identical sets. There are four main phases here. There's prophase, metaphase, anaphase, and telophase that we're going to talk about later on in this lecture. We're going to dive a little deeper into those. But just know that there's four different ones. Then we move on to cytoplasmic division. After the nucleus divides, the rest of the cells need to split too. And that's where this step comes in. It divides, and now we officially have two separate identical cells. And the final stage, differentiation. Once the new cells are formed, they may undergo this process, differentiation, this fourth step, which means the cell becomes specialized by some specific function. So becoming a muscle cell, a nerve cell, blood cell. It's assigned a role, it's assigned a job. This step is really important for cell development. Like we said, all cells have the same DNA, but this is the step that differentiates them. It gives all the cells their unique roles, their unique jobs, and it sends it off to do that job. So now let's break down the structure of a typical human cell. Whether we're talking about skin cells, muscle cells, brain cells, most of them share common parts and functions. There are two main types of cells in the human body, sex cells and somatic cells. Sex cells include sperm and egg cells. They're involved in reproduction and carry only half the genetic material, which is 23 chromosomes. Somatic cells are all the other cells in your body: skin, liver, blood, brain, all of them. These cells have the full 46 chromosomes that make up your organs, muscles, and tissues. So different parts of the cell membrane is the outer layer of the cell. It's like the skin of the cell. It controls what comes in and out. It plays a good role in communication and identifying other cells. Then we have the nucleus, which is the brain or the command center of the cell. It holds DNA, which is your genetic makeup, and controls all cell activities. Inside the nucleus, you'll see the nucleolus, where ribosomes are made, and then you have your cytoplasm. Everything between the nucleus and the membrane is considered your cytoplasm. It's a jelly-like fluid that suspends organelles, and those organelles perform essential jobs like producing energy, building proteins, cleaning up waste, and that's our kind of our basic cell structure, cell makeup. So let's talk more about how the cells divide. There's two main types of cell division. There's meiosis and mitosis, and each has a different purpose and results in a different type of cell. So starting off with meiosis, This process is used to make sex cells, so eggs and females and sperm and males. What makes it so unique is that it reduces the number of chromosomes by half. Like I mentioned on the last slide, it only has 23 chromosomes, which the full number is 46. And when an egg and sperm unite, they create that full 46. So during meiosis, a cell with 46 chromosomes divides in such a way that results in the cells only having 23 chromosomes. These cells are genetically unique, which contributes to the variation we see with siblings and relatives. And that's because of processes like crossing over and independent assortment during meiosis. Moving on to mitosis, this is what most of your body's cells use to divide. Mitosis is the process of nuclear division that leads to the creation of two identical cells. Each cell has the same number of chromosomes as the original, which is 46. And this mitosis process is crucial for growth, repair, and replacement of cells in our bodies. So for example, if you get a cut or you lose skin cells, mitosis kicks in to make new cells and fill in the gaps and heal that scrape on your skin. So while meiosis makes reproductive cells with half of our DNA, 23 chromosomes, mitosis makes body cells that are exact genetic copies of the original. So we've talked about how mitosis is used by most body cells or somatic cells to grow and repair tissue. So let's talk about the 4 major stages of mitosis that I mentioned earlier, and let's dive a little deeper into those. So like we said, mitosis is the process of where a single cell divides to produce two identical cells. It ensures that each cell gets a complete copy of the DNA. And this process happens in almost all cells except sex cells because they go through meiosis, and red blood cells because they don't have a nucleus and they can't divide. So our stages of mitosis, prophase is where the chromosomes condense and they become visible under a microscope. The nuclear membrane dissolves and structures called centrioles start moving to opposite sides of the cell to help guide that division. Then you have metaphase, where the chromosomes line up in the center of the cell. And this alignment is crucial for making sure each new cell gets the right set of chromosomes. It's making sure that everything is distributed the way it should be. Then we have anaphase, where the duplicated chromosomes are pulled apart. Each half is dragged to opposite ends of the cell by spindle fibers. That ensures each new cell will have an identical set of genetic instructions. And then telophase. Telophase is where the cells begin to wrap things up. New nuclear membranes formed around the set of chromosomes, and that starts to unravel again. So mitosis is how we grow, we heal wounds, we replace lost or damaged cells. It's highly controlled, and when it's not, that's when we run into problems like cancer. So understanding each phase helps us recognize how those cells should behave, and it's going to help us understand what happens when they don't. So differentiation, I mentioned this earlier. It's the process where cells become specialized to perform specific tasks. Each cell in your body starts out the same, kind of like a blank slate. And this is how we decide, or this is how the body decides what cell becomes different from one another and what their job is. Even though they all have the same DNA, like we said earlier, cells do different jobs. Muscle cells contract, nerve cells send electrical impulses, blood cells carry oxygen. All these cells started with the same genetic instructions, but differentiation, that step in the cell life cycle, activates the genes needed for each job. So that's how we go from a single fertilized egg to a human made up of over 200 different types of cells. Stem cells are the starting point. These are cells that have the potential to become many types of specialized cells. They can divide over and over without specializing right away, which makes them very useful in development and repair, even medicine, like regenerating damaged tissue. We can think of stem cells as like the master builders, ready to become whatever the body needs, ready to adapt to whatever our body needs it to adapt to. So moving to our cell membrane, also called the plasma membrane, It's one of the most important parts of the cell. It's like the cell's security system, controlling what comes in, what goes out, and how it communicates with the environment. It gives form to our cell. It gives the cell its shape and its structure. It's the outer shell that kind of holds everything inside. And without it, the cell would just collapse or leak. And like we said, it controls movement of substances, so it regulates what enters and exits the cell. It keeps harmful substances out, lets nutrients in, and then it gets rid of waste. This process is also called selective permeability, where the membrane decides what's allowed through. Permeable membrane lets anything pass through, but most cell membranes are semi-permeable, meaning they let some substances through while blocking others. So think of it like a bouncer at a club. Only some things can get through. Only some people can get through that bouncer, not everybody. Within the membrane, there are specialized proteins that do a variety of different jobs. We have channel proteins that form tunnels through the membrane that to allow specific molecules or ions like water or sodium to pass through. They're kind of like doorways that only open for the right size molecules, which is important for maintaining balance inside of the cell. There's enzyme proteins, which speed up chemical reactions happening right at the surface. They may help break down nutrients or build up molecules that the cell needs, and they're often part of a metabolic pathway. We have receptor proteins. They receive chemical signals from outside the cell, like hormones, neurotransmitters, and they trigger a response inside. So, this is how your cells kind of know what's happening in the body and they know how to respond. Identifier proteins serve at like name tags. They help the immune system recognize which cells belong to your body and which don't. This is very important in organ transplants and fighting off infections and keeping you healthy. And then carrier proteins are your transporters. They bind to specific molecules on one side of the membrane and then change shape to move that molecule across. Unlike channel proteins that work like doorways, carrier proteins are more like revolving doors. They're active participants in transports and they just keep turning. All of these proteins together make the cell membrane more than just a barrier. They make it smart, they make it selective, and they make the process interactive. Without these proteins, cells wouldn't be able to communicate, wouldn't be able to transport, and wouldn't be able to protect themselves. So our cytoplasm, you've heard me mention this a few times already. The cytoplasm is a thick jelly-like interior of the cell, and it's kind of where all the action happens. It fills the space between the nucleus and the cell membrane and contains everything the cell needs to stay alive and function properly. It's primarily made of water, but it also contains salts, enzymes, organic molecules. And it's so think of it like the cell's internal ocean. It's a place where all the organelles float and chemical reactions can happen. And it's not just a filler, it's essential for transporting substances and cushioning organelles. Within the cytoplasm, there's membranous networks, which is like the endoplasmic reticulum. There's protein frameworks, which give structure and help with intracellular transport. And then the cytoskeleton, which is a network of protein filaments that help maintain the cell shape and play a role in movement and division. There's two major components. There's a cytosol, which is the fluid portion, the base where everything is suspended, and then there's organelles. These are the cell's internal structures like the mitochondria or ribosomes, each with a specialized job, all working within the cytoplasm. All right, organelles. They have specific actions that help the cell carry out its activities. Like we've said, cells have so many different types of functions. They have so many different jobs. So starting off with our centrioles. Centrioles are involved in cell division. They help organize the spindle fibers that separate chromosomes during mitosis and meiosis. Then we have cilia and flagella. Cilia are small hair-like structures that help move substances across the surface of the cell, kind of like moving mucus in the respiratory tract. And then flagella, like the tail on a sperm cell, is longer and helps the entire cell move. Ribosomes are protein factories. They can be found floating freely within the cytoplasm or attached to the rough endoplasmic reticulum. Their job is to assemble and convert amino acids into proteins. Then you have your endoplasmic reticulum, which there's two different types. There's rough ER and smooth ER. Rough ER has ribosomes on its surface and helps modify and transport proteins. Smooth ER lacks ribosomes and is involved in making lipids and detoxifying substances. The Golgi apparatus is like the shipping and receiving center of the cell. It processes, packages, and sends proteins and lipids to where they're needed, whether that's inside or outside of the cell. Lysosomes are the recycling center. They contain digestive enzymes that break down waste, damage organelles, or harmful bacteria. Microfilaments are part of the cytoskeleton, which helps give the cell structure. And microfilaments are thin and flexible. Same with thick filaments. They're involved in the contraction like myosin and muscle cells, also part of the cytoskeleton. Then you have the mitochondria, which we all know is the powerhouse of the cell. It converts food into usable energy known as ATP through cellular respiration. Cells that need a lot of energy, like muscle cells, have lots of mitochondria. And then we have our vesicles. They're tiny transport bubbles. They carry substances from one part of the cell to the other or to the membrane to release outside of the cell. So it's kind of, the vesicles are kind of like the delivery trucks of our cells. Then we have the nucleus, one of the most important and most recognizable parts of our cell. It's the control center. It manages the cell's activities and houses the genetic material. It's kind of like the city hall. If you were to think of your cell like a small city, nucleus is like the city hall. It's a large, round, oval structure, usually located near the center of the cell, surrounded by nuclear membrane to protect it and control what moves in and out. Kind of like what the cell membrane does, just for the nucleus instead. So like we've said, it contains DNA, the molecule that carries all the instructions for how the cell functions, grows, and reproduces. And it also contains nucleoplasm, which is gel-like fluid on the inside. And the nucleus is essential for cell survival. If it's damaged or missing, the cell can't function properly. There are some cells without a nucleus, like we mentioned earlier, but cells that have a nucleus or need a nucleus can't survive without a nucleus because the cell will just lose its instructions, which is why red blood cells, which are ones that don't have a nucleus, only live for about 120 days. They're think of them like short-term specialists and not long-term managers. So, like I said, the inside of the cell, the way the cells function, can be thought of kind of like being a miniature city. So, on the topic of it being a miniature city,
let's begin with the cell wall. In the city, the cell wall that surrounds the city protects the city from items outside, just like the cell wall provides protection and structure to the cell. Next, the cell membrane is represented by the doors, because the cell membrane is a semi-permeable membrane that regulates what moves in and out of the cell. Next, you can see the cytoplasm in the middle of the city. It is the water found in the city, because the cytoplasm is a gel-like material that contains important nutrients and the other organelles are suspended in. Next up is the nucleus. The nucleus is represented by the city hall because it controls the activities of the cell. Like the nucleus has all the directions and blueprints of the cell because it contains the DNA. The endoplasmic reticulum, or ER, is represented by the roadways of the city because the ER acts like passageways and roadways in order to move materials around the cell. Ribosomes are small factories because the ribosomes are the sites where proteins are made. They are protein factories. The mitochondria are represented by these power plants because the mitochondria generates energy for the cell in the process called cellular respiration. The vacuoles are storage containers because vacuoles store food and water for the cell. The lysosomes are small trash cans because they break down waste material. And finally, the chloroplasts are represented by small solar factories that convert sunlight to energy. I hope this mnemonic device helps for you memorizing the cell organelles.
So cellular signaling. Cells don't operate in isolation. They're constantly talking to each other, communicating, responding about what's happening inside and around them. It's what allows our body to stay alive, balanced, and responsive. So cells use this signaling to do a couple different things. They use it to maintain homeostasis, so they keep our internal conditions stable. They use it to fight infections by recognizing pathogens and coordinating immune responses. They use it to reproduce through hormone signals, perform normal functions like contraction, muscle contraction, digestion. And the way this happens is through chemical signals like hormones, which are used for long distance messaging, neurotransmitters, which are fast signals between nerve cells, and then local signals like immune cells calling for help at the site of the injury. So when immune cells need help fighting off that infection, that's a local signal they send out for more immune cells to come and help fight off the infection. Receptor cells or receptor proteins on the cell membrane recognize these signals and they trigger changes inside the cell. So it's kind of like receiving a phone call. The receptor is the phone and the message tells the cell what to do. Even inside a single cell, signaling helps coordinate processes. For example, the nucleus may send instructions to the mitochondria to increase energy output. Now, when the signals are too weak, too strong, or misinterpreted, it can lead to problems. And it can lead to problems like cancer. Cancer can occur if the signals that tell a cell to stop dividing are ignored. Diabetes can occur if cells don't respond properly to insulin. Autoimmune diseases happen when immune signals misidentify your own cells as threats. So, just like in society, communication is key. Communication solves 98% of problems, and that goes for cellular signaling as well. When cells signal properly, everything runs smoothly. When signals are disrupted, it can lead to dysfunction and disease. Cellular respiration, this is the process where cells convert nutrients, typically primarily glucose, into usable energy in a form called adenosine triphosphate or ATP. This energy is essential for all cellular functions from muscle contractions to nerve signaling. And the process occurs in three main stages: glycolysis, the Krebs cycle, and electron transport chain. So the first step is glycolysis, which takes place in the cytoplasm outside the mitochondria. Glucose is broken down into carbon molecules of pyruvic acid. This process does not require oxygen, so it's considered anaerobic, and it uses a small amount of ATP. And this step is like the cell's emergency generator. It works fast even without oxygen. Then the next step is the Krebs cycle, where a few more ATP molecules are produced. If oxygen is present, pyruvic acid enters the mitochondria and then goes through this step, the Krebs cycle. More energy is extracted as carbon dioxide is released. And most importantly, this cycle generates high energy electron carriers for the next step of electron transport system. This is the most productive stage. It also occurs in the mitochondria. And it's where those electrons from the Krebs cycle are passed down a chain of proteins. They move along, they help pump protons and create a gradient that powers that enzyme ATP. And this step produces the bulk of energy. This is kind of where the cell cashes in on everything it's been building up. So why is this all important, right? Why are you sitting here listening to me talk about cells and homeostasis and not about how to treat somebody's injury or treat somebody's illness, right? So we need to build a foundational understanding of human biology so that we can add to it when you start learning about trauma, cardiac, neuro, all of these things you're going to be treating as paramedics. It's not good enough to just know that something's happening. You have to know the why behind it. The why is going to help you decide what to do for that patient using critical thinking. Any medical condition you encounter starts at a cellular level, whether the cells are responding to infection, whether they're damaged by toxins, whether they're mutating. These all present as medical issues that you will run into in your career. And your treatment as paramedics ultimately aims to correct cellular dysfunction. So, for example, you may just say you're administering glucose to your hyperglycemic patient because their sugar's low, but what you're actually doing is repairing cellular function by providing the necessary energy source, which is sugar, for cellular metabolism. So, this whole lecture has been pretty science heavy, but the bottom line is that every sign, symptom, or complaint we see in EMS is the result of something happening at the cellular level. Whether it's chest pain from a lack of oxygen to cardiac cells, altered mental status from electrolyte imbalances, fever from immune cells releasing inflammatory signals, it all comes down to our cells functioning or not functioning. So, again, what does this have to do with EMS? Great. I know that if somebody has chest pain, it could be from a lack of oxygen to cardiac cells. Great. Awesome. But why else does this have to do with EMS? Well, let's start with this first photo. This first photo, this patient's having an allergic reaction to what looks like grapefruit. And in allergic reactions, in anaphylaxis, our mast cells play a big role. When exposed to an allergen, these cells release histamine, which causes fluids to move into our tissues. And this causes swelling and inflammation that we see on assessment, and that can negatively impact the function of vital organs. For example, in anaphylaxis, we're worried about the airway swelling shut. That's one of our biggest concerns when it comes to anaphylaxis. So not only are we worried about the swelling and inflammation, but as fluid leaves the bloodstream and goes into the tissues, less blood is available to circulate oxygen and nutrients, which then negatively impacts perfusion. With less blood available, our sympathetic nervous system gets activated and our patient becomes hypotensive. Then they will become tachycardic because their heart is trying to maintain adequate cardiac output. Now, as paramedics, our job is to correct this cellular dysfunction that has caused all these symptoms that we see on our assessment, to correct the swelling, to correct the hypotension, to help to correct the tachycardia. So all of these signs and symptoms we see on our anaphylactic patients was from a cellular dysfunction. All right, photo number 2. This guy looks like he's having chest pain. When there's an insufficient amount of oxygen in the cardiac cells in the heart, there's a reduction of ATP production, which impairs the heart's ability to function properly. ATP is the main energy source, main energy currency in the cell, and a lack of it, or a reduction of ATP production, can lead to myocardial ischemia, which is a partial blockage to the heart that we as paramedics may see on an EKG. This lack of oxygen activates nerve fibers in the heart, which then transmit pain signals to the brain, resulting in the chest pain that our patient is feeling. And then our last photo, he looks like he's going to throw up, but he's supposed to have the flu. So the influenza virus binds to receptors on the surface of cells in the respiratory tract. The virus then enters the cell and begins to get replicated. In response to this, the body's immune system is activated, and the body uses defense mechanisms to allow immune cells easier access to the site of infection. Due to the permeability of those blood vessels, fluid leakage occurs, or can occur, and then contributes to symptoms like congestion, coughing, and difficulty breathing. So even something as minor as the flu, because a lot of times, especially for young people, the flu is pretty minor, can be stripped down to a cellular level. So even though all these chief complaints are different, they all reflect underlying cellular dysfunction. And that's why it's so important for us as clinicians to understand what's going on at this level, to understand the most basic level of this A&P, so that we can understand why we're seeing what we see and why we're doing what we're doing to treat it. So to bring it all home, all living things are made of cells, the smallest unit of life. Cells perform those essential functions that we talked about. Cell structures play a very specific role in keeping us functioning. Cell communication is crucial for us to stay healthy and alive. Cellular respiration powers everything our cells do, and oxygen is critical to that process. Everything we see in EMS, every symptom we treat, comes down to something happening at the cellular level, whether it's a lack of oxygen, an immune response, or an infection. It's all rooted to how our cells are functioning or failing to function. So that's all I have for you about the cell. Thank you and good luck on the rest of your paramedic journey.